So good evening and good morning, good afternoon, everyone. Welcome to the ISAC webinar today. Today's webinar is on antimicrobial pharmacometrics, mathematical model for antimicrobial research and development. And my name is Jinxin Zhao, and I'm a research fellow at the Monash University, Melbourne, Australia. So before we start today's webinar, I would like to introduce you to the ISAC and ISAC early career. The International Society of Antimicrobial Chemotherapy is a major society aimed at advancing the education and science of therapy of infection through establishment of an international federation of affiliated national and regional societies with similar aim and objectives. ISAC currently has more than 90 member societies and over 50,000 individual members. If you are interested and need more information, feel free to get in touch with us. Under the ISAC, there are 21 working groups. The early career working group that I'm a part of works closely with the ISAC and other ECR members, focused on providing a globally accessible platform to communicate with senior researchers and build long-lasting networks for collaborations. Our working group leadership team are actively working with ISAC and other ECR members to propose programs and events for the ECR community. New ideas are always welcome. Please do scan the QR code if you would like to join the team. I would also like to bring your attention to the ICC conference at the end of this year in Perth, Australia. There's an interesting lineup of ECR events in this conference with the support from the ISAC. If you plan to attend any conference this year, do consider this one. And back to the webinar. So the aim of this webinar series is to introduce the latest research and application of pharmacometrics and systems pharmacology in the field of infectious disease. We have a great lineup of top scientists from around the world as confirmed speaker in this series, and we look forward to meeting you all in the future webinars as well. We will also host hands-on session in the future. Now allow me to introduce our speaker, Dr. Yu-Wei. Dr. Lin is an Associate Director of Pharmacometrics at Setara and the co-director of the Malaya Translational and Clinical Pharmacometrics Group. Dr. Lin has over 7 years of academic research experience in antimicrobial pharmacokinetics, pharmacodynamics, and toxicodynamics and system pharmacology. Dr. Lin has a very impressive track record with 42 publications in antimicrobial research, as well as several awards, fellowships, and research grants that include ASCAPT Investigator Award. He is also an invited guest editor for several journals, including the International Journal of Antimicrobial Agents, Frontiers in Pharmacology, and scientific advisor to the Journal of Pharmaceutical Science, and serves as a member of the IQ Consortium Pediatric PBPK Working Group. Now I'm going to hand over to you, Dr. Yu-Wei.
So thank you so much, Jason, for your kind introductions, and good morning, good afternoon, or good evening, everyone. Thank you all for taking the time to be here today. So the topic of today's webinar will be on antimicrobial pharmacometrics, mathematical models for antimicrobial research and development. So I would like to start off my presentation today by asking everyone a question related to ciprofloxacin. Most of us would have fallen sick in the past, and many of us would have taken antibiotics for many different types of infection. Have any of you actually thought how antibiotic dosing regimens are optimized? How do the doctors decide on the dosing regimens? Do they just randomly pick a dose depending on his or her mood or out of his or her own personal experience? I really hope this is not the case. So the antibiotic dosing regimens are optimized using pharmacokinetics and pharmacodynamic studies. So let us look at this famous host-pathogen drug interaction figure where we have the drug represents antibiotics, and we have the box which represents the bacterial, as well as the host which represents humans. The host which represents humans. So over the past decade, antibiotic dosing regimens were optimized based on PK, pharmacokinetics, PD, pharmacodynamic, as well as TD, toxicodynamics, where PK is what the body does to the antibiotics, and PD is what the drug does to the bacterial, and TD is the associated side effect. So dosing regimen is dependent on the optimal balance of the three. The dose that we give will have to give sufficiently high exposures to kill the bacterials, but not so high to cause severe toxicity. For antibiotics, the dose will also have to be specifically designed such that it minimizes the development of bacterial resistance. So how do we balance these three individual components to rationally design dosing regimens or to rationally design a novel antibiotics? This, of course, can be done through pharmacometrics. So what is pharmacometrics? Pharmacometrics is the sciences of developing and applying mathematical and statistical models to characterize, understand, and to predict a drug's pharmacokinetics, pharmacodynamic, as well as toxicodynamics. Pharmacometrics, also known as quantitative pharmacology or population PK/PD modeling, has been widely utilized in drug development and fully recognized by the global regulatory authority, such as FDA and EMA, as well as PMDA. The goal of today's webinar is to introduce you all to different pharmacometric techniques and highlight using examples, showing you how pharmacometrics can be utilized in antimicrobial research and development. So I have divided the presentations into 3 or 4 parts. So I'll first give brief introductions to population PK modeling, and then give you a quick examples to demonstrate how these models were used to set dosing guidance for polymyxin B. Then I will introduce empirical PD, as well as mechanism-based PK/PD modeling, and share 2 examples of how we apply this modeling technique for our polymyxin-based double and triple combination therapy. Finally, I will highlight the utilities and usefulness of pharmacometrics by sharing examples of how these models can be used to translationally optimize regimens in humans, in other words, how we use these models to bridge the gap between in vitro and in vivo studies. So the goal of population PK/PD modeling or pharmacometrics is essentially to describe the relationship between the dosing regimen, as well as the plasma concentrations, the exposures, as well as the toxicities, as well as safety, as well as efficacy endpoint. In all this relationship, of course, there are some sort of variabilities between different subjects. For example, if you give the same dosing regimens to 2 different subjects, you will give rise to 2 different concentration time profiles, and of course that will result in 2 different safety as well as efficacy endpoint. Even if both patients had the same concentration curve, that same concentrations could also potentially give rise to 2 different safety as well as efficacy outcome. We can quantify this variability using pharmacometrics, as well as identifying factors or predictors that contribute or explain these differences between subjects. Once the relationship are established, we can use this relationship to conduct in silico clinical trial simulations and propose or even suggest an optimal regimen considering both the PK, PD, as well as TD. Today, we will only focus on the PK/PD aspect. So let us look at how we can analyze PK in the pharmacometric world. So there are 3 different approaches that we can use to analyze PK data. The first approach is the naive pool data analysis, where we ignore all variabilities from subjects. The second is the two-stage approach, and the last being the population approach, which is pretty much the gold standard proposed in the 1990s. So naive pool analysis, as the name implies, it ignores the variability and assumes all data arise from a single individual. Each individual line in this graph represents one individual. It can be humans, it can be rats, it can be monkeys. Naive pool analysis essentially just fit one line, and for all subjects, they all have one clearance and volume distributions parameter. This approach is probably the most widely used approach in non-clinical studies because of the nature of the data, because we often use destructive sampling in non-clinical studies. And it's also because this method is very easy to implement compared to the population approach. For non-clinical study, especially for lower species, such as mice, because destructive sampling is often used, this bias, we have to put up with the bias because there's no other way around it. However, for higher animal species, or even for clinical PK studies, naive pool analysis should be avoided if all possible. So let me give you an example of why this should be avoided. Each data point here represents data from different individual. The black line here is the model predictions from the naive pool analysis. The red dots here are data from a particular individual, and you can see that these subjects have a PK profile quite different to the solid black line. So if we were to use the clearance and volume distributions parameters derived from this solid black line, the naive pool analysis, to optimize the dosing regimens, then we most likely will not dose these subjects correctly because the PK profile is very different and has a much shorter half-life than the general population. The second most commonly used approach in both non-clinical and clinical space is the standard two-stage approach. This is where you determine the PK parameters for each subject or animal independently in stage 1, and then in stage 2, you perform descriptive statistics to derive the mean and standard deviations. So that's what I'm showing in his table here. For each subject, you derive its own individual PK parameters. Then you do a very simple descriptive statistic calculations to determine the mean as well as standard deviation. The standard two-stage approach is probably the most commonly used approach in a non-clinical space because many researchers are often only interested in determining the primary PK parameters and check for dose linearity. One major drawback of this approach is that it is difficult to use this approach to identify source of variability, and hence it may be difficult to use this to guide or individualize dosing regimen. The last and the population approach is perhaps the best approach out of the three, as it estimates all subjects simultaneously using nonlinear mixed effect modeling. Population PK analysis is the gold standard in clinical study, and it has also been increasingly utilized in a non-clinical space. So by using population PK analysis, it can greatly reduce the numbers of samples you need per animals or humans. So let me go through the general structures, typical structures of a population PK model. It usually consists of 3 different components. So it has the structure component that determines the shape of the distributions or the kinetic profile, whether the drug follows a 1-compartment model kinetics or a 2-compartment model kinetic. It has a variability model component that determines how variable the PK profiles are between subjects or between occasions, or as well as uncertainty associated with the observations. Finally, we have the covariant models, where we try to find variable that can explain the variability and potentially be used to individualize the dose. I will later give you an example of how we utilize covariant modeling to design dosing regimens for polymyxin B. So I wanted to give you a graphical illustrations of each of the components that I just described. So this is an illustration of the a one compartment model following intravenous bolus administrations. So we have the concentration time profile here. So the red line here are essentially model predictions, a model predicted concentration time profile across 24 hours. The dots, of course, are the actual observation. The shape of this model predictions is determined by the PK, the structural model. It's a one compartment model. That's why it's resembling a one compartment model shape. If you have a two compartment models, then you would have a distribution phase followed by a terminal elimination phase. Typically, in a clinical trials or when you do animal PK studies, hopefully you will have more than one individual. And of course, each individual will most likely give you a different PK profile as a result of differences in clearance of volume distribution. In the population PK modeling, we can then estimate the population median, which is the solid black line here, as well as the PK profile for each individual represents for different color lines here simultaneously. So to fully understand the concept, we have to look at this figure here. Here we have a typical concentration time profile with the concentrations on the y-axis and time on the x-axis. So the solid red line is the population median, and the blue line here is the individual concentration time profile of a particular individual. So in simple English, what the population PK modeling is doing is essentially quantifying the difference between the population median as well as the blue line, which is the individual concentration time profile. And that inter-individual differences is called the eta. After you correct for that inter-individual differences, you see that the blue line and the actual observations, there are still some discrepancy. This differences is called epsilon. So what population PK modeling is doing is essentially, in simple English, estimating the fixed effect, the red line, as well as the variability, as well as estimating the variability component simultaneously. And I hope now you can see that population PK modeling is extremely powerful, because once you know the shape of the distributions, which we always assume, it's no, well, most of the case we assume, most of the time we assume is normal distributions. We know the spread of these distributions. We know the population medians. Then we can use those relationships to conduct virtual clinical trial simulations, because we know how clearance as well as the error in our data are distributed. And I'll later give you an examples of how we use in silico clinical trial simulations to optimize dosing regimens and guide dosing regimens. After we have developed the models must be checked and verified before we can use them for future applications. In pharmacometrics, we have different tools that we can use to perform model evaluation. So here we have, I guess, a typical goodness of fit plot on the left, where we plot the model predictions. So on the x-axis against the actual observations. So if the model describes the data very well, of course, then you would assume you would expect that the dots will line up along a line of unity, because predictions would equals to observations 100%. And if that's the case, you would expect that the dots to line up along the line of unity perfectly. On the right, we have what we call a simulation-based diagnostics. So the graph looks extremely complex, but it's actually very, very straightforward to understand the graph, because it is essentially just an overlay of the model predictions versus the actual observations. What we want to see is that if the model describes the data well, then you would expect that the model predictions to line up with the observations. And in this case, you can see that our model, or this particular model is taken from this paper, is describing the central tendency of the data very well as describing the variability, the spread of the data very quite well as well. As you can see that the observations as well as the model predictions line up reasonably well. If you want to learn more about pharmacometric model evaluation techniques, then I suggest you can have a read of this paper, or stay tuned to our future webinar. So, when is population PK modeling useful? It's always useful. It is particularly useful when you have sparse PK sampling. When we have very sparse PK sampling, of course, we cannot analyze the data using a non-compartmental analysis. But with population PK modeling, we can still analyze sparse PK samples. Population PK modeling is particularly useful if you want to learn about the source of variability. And I will talk more about this in the following slide. Finally, population PK modeling, of course, is a hypothesis generating tool so that you can use them to design future studies in both clinical and non-clinical space. And in particular, population PK modeling is extremely useful in pediatric drug development because you can use the adult population PK model to design the sample size as well as sampling points for your pediatric clinical trials, and to maximize the success rate and to reduce the cost of your pediatric clinical trials. And I will later show you an examples of how we did this, and in real life drug development program. So now I want to use the case studies from Professor Zhang Li's lab, polymixing B, as an example to show you the utilities of polymixing of population PK model in dose optimizations, or in guiding dosing regimen. So a population PK model was developed for polymixing B for 24 patients to quantify the PK profiles as well as the variability between subjects. Covariate modeling was performed to identify factors that is causing the variability in the PK profiles. So in other words, understanding why there are differences in clearance as well as volume of distribution parameters between different individuals. Total body weight was identified as a statistically significant covariate. And in other words, so different body weights will result in different PK profile, and is likely the reasons why we have different PK profiles across different individuals. Creatinine clearance was also tested. However, it was not a statistically significant covariate in this particular population PK model. So the findings were then translated and implemented in the polymixing B guidance. So here we have a guidance published from Professor Zhang Li's group at Monash University. And you can see that because total body weight was not identified as a significant covariate, polymixing B, of course, is recommended. Because total body weight was identified as a significant covariate, therefore polymixing B is recommended to administer using a weight-based dosing regimen. Creatinine clearance was not a significant covariate, therefore the dosing regimen does not need to be adjusted for patients with renal impairment. Here I included additional examples of the utilities of the population PK model in antimicrobial research and developments. And of course, I also included examples in pediatric setting as well, done by one of my colleagues in Satara, where we utilized population PK models to help design experiments, clinical trials for pediatrics clinical trials, developments for antimicrobial chemotherapy. So now we will move on to move from population PK modelings to mathematical models that describe or link the PK/PD relationship. So before getting into the mathematical models, I just wanted to briefly touch on the experimental setup for antimicrobial research and development. So coming back to this famous figure that I showed earlier in my presentations, PK/PD modeling is essentially using mathematical model to link PK to PD. But before we can actually build any model to describe this relationship, we need to have data, right? If we don't have data, how can we even link PK to PD? So in the antimicrobial world, this PK and PD relationship can be studied in a test tube. So static time cube experiment is is a very simple experimental setting, and is perhaps one of the simplest experimental settings we have in the antimicrobial world. So as the name implies, in time cube experiments, we follow the growth and killing of the bacteria caused by antibiotic over time. So for people that don't know what static time cube experiment is, we have different test tubes there. So with bacteria with the same initial inoculum, and we add antibiotics in an increasing concentration. So for example, this one could be a growth control with bacteria only. This one we have add polymyxin B 1 milligrams per liter, 2 milligrams per liter, and then 4 milligrams per liter. Then we take samples at various time points across 24 or 48 hours, and then quantify the bacterial count. Static time cube experiment is relatively easy to set up. And of course, it can cover a wide range of concentration to allow us to build a model to quantify the concentrations versus bacterial killing over time. However, there are limitations with this setup. Firstly, because the concentration is static. Firstly, the concentration is static. That means that concentration does not fluctuate over time. And because of this, the media is actually not replenished, therefore the duration of the experiment is often limited to either 24 or 48 hours. To overcome these limitations, in the antimicrobial world, we have something called, dynamic systems. So we have either the in vitro one-compartment models or the hollow fiber infection models. In these systems, the media is replenished, and the study durations can therefore be extended beyond 48 hours. Most importantly, in this dynamic systems, the PK profile is no longer static. So we can use this system to simulate concentration time profile to mimic real human PK profiles following treatment with clinically feasible dosing regimens. However, there's one major drawback for this experiment, and that is they are very expensive, they are time-consuming, as well as labor-consuming to set up and perform. So enough of the the script talks on the experimental setup. So once we generated data, now we have the data. So of course, we're going to build a model to describe that relationship. So this is perhaps the most exciting part of antimicrobial and pharmacometrics. So this is where we build mathematical model and use mathematical model as a tool to digitize the data that you generated in vitro static time cubes experiments or in vitro dynamic model experiments. You transform those data into mathematical expressions so that we can use them for translational purposes. So typically, PK/PD model consists of 3 different components. So we have a bacterial component that describes the growth and death of the bacteria. We have a drug component or antibiotic component that describes the kinetics of the drug in a test tube or in humans. Then finally, we have, another component, which is the antibiotic pathogen component that describes the interactions between the drug as well as the bacteria. So starting with the bacterial model, there are many different empirical models available to describe the bacterial growth without the presence of drug. In pharmacometrics, well, actually, there are more than 3, but in pharmacometrics, I guess this 3 are the probably most utilized one. So exponential growth model, of course, is one of the simplest, which consists of first of the growth rate parameters as well as the natural death rate parameters. As the name implies, this mathematical model can only describe the exponential part of the models, and this model is therefore unable to describe the plateauing of the bacterial count as you approach 24 or 48 hours. These limitations, of course, can be easily overcome by using logistic growth models. This is where the R represents the growth rate. And at time 0, of course, the growth rate is going to be at its maximum because that's assumed the numbers of bacterial count at time 0 will be quite small. And as the count grows, it approaches the maximum carrying capacity of the systems. So this will approach K, and divided by K equals to 1, because n equals to K. 1 minus 1 equals to 0. So that's why this logistic growth equations and then growth rate approaches 0. And that's how the curve approaches a plateau. Another empirical model that is commonly used is the capacity rate limited growth model, of course, first proposed by Professor Alan Frost from the University of North Carolina. So this first part of the equations is essentially a Michaelis-Menten equations that control, I guess, the shape of this curve. And the second part is basically just the natural death rate of the bacteria. In addition to those empirical models, there are two different types of mechanism-based PKPD model that we can also utilize to describe the bacterial growth. The first one is called the persistent state model, and second is called the life cycle model. The persistent state model, proposed by the Uppsala team, describe the phenotypical switch of the bacterial. So you have state 1, which is the bacterials in the growing stage. Then you have switching to state 2, which is the non-growing state. The life cycle models, on the other hand, describe the life cycle of a particular bacterial cell. So we again have two states. However, this time state 1 represents the vegetative state, where the bacterial cell is getting ready to replicate. And state 2 represents the replicated state. That is the state where the bacterial cells start to replicate and double in publications. As you can see that both models are very different to the empirical PD model, as both empirical PD model, because both of these models are based on physiological processes of bacterial replication or system. So over to the second component, which is the drug model. So this model, I already briefly mentioned about this one in the in vivo systems, essentially describe the change in antibiotic concentrations over time. In a static time to experiment, as I already mentioned, that's where you add drug directly into this test tube. There is essentially no eliminations of the drug. So the concentrations, in theory, should remain constant. However, this is often not the case. For many drugs like polymyxin, will likely stick to the labware or plasticware or even bind to the LPS, and this will cause a decrease in concentrations. And therefore, the concentrations for the static time to experiments will actually decrease over time. They won't remain constant. Therefore, it is also very important to quantify the drug concentrations over time so that you can have this rate of decrease of degradations implemented into your models for the static time to experiment. In the dynamic time to experiment, however, the rate of eliminations, of course, is set manually by the experimenters. So you have a pump that is constantly pumping and eliminating the drug out of the system. So you can essentially set the elimination rate to the pump flow rate. However, it is also very important to actually quantify the concentration, because drugs like polymyxins will also bind to plasticware, bind to LPS, and that will also cause a decrease in concentrations. So it's also important to account for those differences. Finally, in the animal infection model, the eliminations of the drug will be determined based on the PK/PD study as described in part 1 of this webinar. So the last part of the PK/PD model is the antibiotic pathogen interaction model. The drug effect can be added to either inhibit the growth or to stimulate the killing. The drug effect can be parameterized using different mathematical expressions. So commonly we use either the slope, the power, the Emax, or the sigmoidal Emax equation. Which of these parameterizations we use really depend on the amount. It's really dependent on the amount of data as well as the quality of the data that we have, because pharmacometric is a data-driven model. So in summary, when you put everything together, and if you wanted to use a persistent state model, you would get this is a basic schematic of the persistent state model that you could potentially use to describe the killing of a monotherapy. So this is the typical predictions that I borrowed from a Nielsen's review paper for this particular model. And if you are an antimicrobial pharmacologist, one thing you will notice straight away is that the shape of this killing doesn't actually reflect the real life time-Q experiment. So here we have, here I have included, a real life time-Q experiment, in-house data from the Lee's lab. This is polymixing B against a particular isolate against a wide range of concentrations. So we have the growth controls and then 8 times the or 8 milligrams per liters concentrations. You can see that at 24 hours we have regrowth. However, if you look at this model predictions, you see that there's essentially no regrowth. So in real life, unfortunately, in real life, when you do time-Q experiments or dynamic time-Q experiments, what you often see is regrowth. And this regrowth can be due to a lot of different things. But one of the major causes of this regrowth, of course, is due to resistance. So the question is, can PKPD model be used to describe the regrowth observed in a time-Q experiment? And of course, it's a yes. To account for the resistance, we have 2 different types of models. So the most commonly used one is perhaps the heteroresistant model, where we assume several pre-existing subpopulations, and each will have different drug susceptibility. Regrowth is essentially due to the preferential killing of the most susceptible subpopulation. The other model is the adaptive resistant model, which is the reversible form of resistance, which is concentration dependent. Adaptive resistance in this case is often modeled using what we call a binding functions. So at time 0, we usually assume that resistance is, is completely off. As the concentration increases, that we switch on the resistance, and with increasing resistance, that decrease the antimicrobial efficacies, and that of course causes a decrease in killing of the bacterial. And after inclusions of either of this, you can see that the model predictions is now able to describe the regrowth that we typically observe in a real life setting. So finally, I just wanted to show you how we can use PKPD modeling for antibiotic double and triple combination therapy. Here is an illustration for polymixing based double and double combination therapy. This is the model structure used for this double combination therapy. So so the bacterial model is a persistent state model with 2 pre-existing subpopulations. Polymixing B and rifampicin monotherapy was assumed to kill the susceptible bacteria by the slope function. In combinations, polymixing B was assumed to enhance the rifampicin uptake and hence increase the antimicrobial efficacies of rifampicin. One interesting part, one interesting thing about this model is that MIC was also included as a covariate in the model, so that this model is able to predict killing of mono and combination therapy against isolates of different MICs. Here I'm showing you model predictions or model evaluation tools that visual predictive checks that I mentioned early on in my presentations today. So I'm only showing a result for 2 strands. Here we have the rifampicins, and here we have the polymixing B. So this is polymixing B0 all the way to polymyxin B8, and this is a refined percent 0 all the way to refined percent 32. The shaded area represents the prediction intervals or model predictions, while the red circles represent observations. And you can see that our model describes the data very well across isolates of different MIC. PK/PD model can also be applied to triple combination therapy. For this particular model, we use the capacity limited growth model with 4 pre-existing subpopulations for our polymyxin B, amikacin, and rifampicin triple combination therapy. In these particular combinations, polymyxin B was assumed to enhance the uptake of amikacin as well as rifampicin. At the same time, amikacin was also assumed to enhance the uptake of rifampicin. To describe the observed synergies for this triple combination therapy, subpopulation synergy was implemented in addition to the mechanistic synergy described here. So again, here is our model assessment, and you can see that our model describes the data very well across a wide range of concentrations. So here we have different concentrations for double combination therapy, and here we have different concentrations for the triple combination therapy. PK/PD modeling, of course, can also be applied to non-antibiotic combination therapy, such as phage and antibiotic combination therapy. So for this particular model, the author actually included 2 subpopulations, one phage sensitive, as well as another being an antibiotic resistant subpopulations. Phage is assumed as a monotherapy, assumed to replicate within the host and decay randomly following a first order decay rate constant. Ciprofloxacin was assumed to kill the antibiotic sensitivity bacteria as a monotherapy. In combinations, the antimicrobial synergy was assumed to be a result of the subpopulation synergy. So in other words, the phage-resistant subpopulations was killed by ciprofloxacin, while the phage-susceptible subpopulations was killed by phage, and vice versa. So, and what is unique about this model compared to the 3 other models that I showed early on is that for this particular model, you see we also have a host component. So that this particular author also included, immune system into this model. So now the model is able to predict the likely PK as well as PD profiles following treatments with ciprofloxacin as well as phage therapy in both immunocompromised as well as immunocompetent patients. And you can see how powerful it is, and I will show you how powerful it is. This can be in the next section. So now I will describe how we can combine this PK/PD models with humans or animal mouse PK models for translations. So I'll be using the triple combination therapy as an example. So in order to translate the models from test tube, to translate the data from test tube into humans, we can link the PK model developed in vitro to the human population PK model developed based on the human PK data. So in the population PK models, if you can recall, we established the relationship between the dose and concentrations. At the same time, we also quantify the variability between different individuals. We can then use this model to simulate large numbers of virtual patients because we know how clearance and volume distributions are distributed within a particular population. In the PK/PD model, on the other hand, we know the relationship between the concentrations and the bacterial load. So therefore, by linking the PK profiles from the virtual populations to the PD profiles up to the PK/PD model generated based on the in vitro time-cube studies, we can predict the likely PD profile for each virtual subjects. So now you can see how powerful it is. it's essentially an in silico clinical trial simulations. So one thing we need to be aware of is that the bacteria, of course, will grow differently in the in vitro systems as well compared to the in vivo systems, right? Bacteria obviously will not grow at the same speed or the same rate when they are in broth as opposed to when they're in blood. So therefore, we have to correct for those differences before we can translate the findings from test tube into humans. So this we usually adjust for the growth rate as well as the maximum carry capacity using data generated from the animal infection model. And this is exactly what we did for our triple combination therapy. So here we are. So we took our triple combination therapy models that I showed you early on. We combine with the human population PK models from the literatures. Then we simulate different clinically achievable dosing regimens. We conduct a very large size clinical trials around 10,000 subjects. And then we predict the likely PD profiles for each of these projects, each of the subjects based on our PK/PD modeling results. So here we are. Let's look at the polymyxin B monotherapy result first. So this is essentially, I guess, the killing curve in humans for this 10,000 subjects. You can see that following treatment with polymyxin B monotherapy, the drug is essentially ineffective. As you can see, it started at 6 log CFU per ml, and with time, it essentially increased to 48. Even with the maximum highest possible dose, so that should be 15, not 1.5, you see that the bacteria did not even respond to the therapy. So, and I'm not surprised because this isolate that we use is essentially with a polymyxin B MIC of I think greater than 32 or so. So I'm not surprised that monotherapy is not very effective or is not effective at all. So here we have the amikacin therapy, amikacin monotherapy, and you can see that we can see a slow decrease, a drop in the killing in the bacterial load, however, followed by regrowth. For second treatment, it dropped, but eventually the therapy is ineffective against this particular isolate, either for the low dose or for the high dose. Then moving on to rifampicin, again, you see that there's a drop in the therapy and you see a bit of variability. So some patients do see a greater drop compared to other patients. At the green line, which is the medium, you see that majority of the patients do have a very, very tiny drop, but eventually regrowth happens and the drug is completely, the bacterial is completely resistant to the antibiotics. Then we have the combination therapy. You see that out of the 3 possible combinations, amikacin plus rifampicin and polymyxin plus rifampicin are effective only at the first dose. You see a last drop, but then eventually a regrowth. And the result, of course, is quite consistent across this 10,000 virtual subjects. However, when you look at the polymyxin plus amikacin double combination therapy, you see this large drop. And eventually, at the medium of this population, you see that most of them are not very effective. However, you do see some portion of these patients, of this virtual populations, being responsive to this double combination therapy. So this one is probably not as effective as this two, but the 3 of them are not effective, but this one is perhaps could be effective in some populations or in some patients. So now moving on to the triple combination therapy, you see that we have, the maximum possible dose as well as the lowest possible dose, and you see that our triple combination therapy could be effective if we use one of a low dose of polymyxin B plus 2 high dose of amikacin and rifampicin. At the 50, at the 50% prediction at the 50 percentiles, and you see that we could potentially eliminate this particular isolate at 48 hours. For this dose, however, you see that there are still large portions of these populations not responsive to this particular dosing regimens. So based on the PK/PD modeling simulation result, then you can make judgment. If you were to bring these combinations into clinical trials, which dose would you pick, and what are the likely chance of you seeing a positive clinical outcome? So, and now hopefully you can see how powerful PK/PD modeling can be in designing clinical trials, as well as selecting your, a dose for your clinical trials to maximize the success rate, as well as minimize the failure rate. So in summary, so this is the proposed antimicrobial model-informed drug development pipeline. So once you have a compound in mind, a lead candidate, what you should do is you should start with static time-Q experiment. Then you would use those results generated from your static time-Q experiments to develop a PK/PD model. Then you would use the developed models to basically design your IBM as well as your hollow fiber experiments. Why do we have to do this? Because those experiments are extremely expensive. Therefore, we want to use our developed model to minimize, I guess, the numbers of experiments that we have to perform for IBM as well as hollow fiber experiments. Once we have the IBM and hollow fiber experiments generated, we would then use those models to validate our model. If the model fit well, that's good. If the model doesn't fit and does not describe the data, then we will incorporate those data into our model to refine our model. Once we have a model that describes the hollow fiber and IBM data well, we then use those refined PK/PD model to design animal experiments. Then again, we'll repeat the process again. We use the data generated in the animal infection models to validate the models or to refine the models. Then the last step, of course, is translations into humans. If this is a new antibiotics, then what you would do is, of course, you would then allometrically scale the PK from animals to humans, and then use your PK/PD model to predict the likely killing profile for different clinically achievable dosing regimens. So you can use this PK/PD model to select, I guess, a first-in-human dose to maximize the success rate of your clinical trial. If it's an existing drug, so similar to the examples that I showed using a triple combination therapies, where you are optimizing, dosing regimens, then you would borrow the population PK models from the literatures, merge it with your mechanism-based PK/PD model, then perform simulations to optimize your dosing regimens in humans, and you can do that for different subpopulations as well. You can do that in pediatrics, do that in people with hepatic or renal impairments subjects. So each of this step is essentially a learning and confirming curve. So you use your data, you teach your models to learn, you then use those to generate new, perform simulations, then you do experiments, you confirm them, and then you go back to learning. So it's a learning and confirming curve. So the last sections I want to briefly touch on before I conclude my talk today is I want to give you guys a few more examples of antimicrobial pharmacometrics for research and development. So pharmacometrics have, and can also be used in different areas as well. So, and this is a very good example I want to share is that a research group from Leiden, they have actually applied pharmacometric principles to design a collateral sensitivity-based dosing regimens. They are also increasing, use of integrations of multi-omics with PK/PD modeling to help with precision dosing. Finally, pharmacometrics, of course, is not only applicable to research and development. Pharmacometrics is also useful in clinical settings. So there is a few which is called what they call a model-informed precision dosing. So it's also in dose and by the infectious disease societies as well. So the one I'm including here is an example of vancomycin. So you can see that the guidance essentially suggests that using a model-informed precision dosing, so a Bayesian software program, is a preferred approach for dose optimizations. So pharmacometrics is also very, very useful for clinical settings. So where you can take samples from subjects, put that into the models, and then the models will then give you an individualized clearance and volume distributions for that particular individuals. Then you can use those individualized clearance, as well as volume distributions to derive an individualized dose for that particular subject. So that's, in my mind, that's a much better approach than a one dose fit all approach. So thank you very much for your attention. And with that, I'm happy to take any questions that you may have.
Sure, thanks, Dr. Yu-Wei. We actually have quite a few questions already posted in the Q&A session, and because we are actually a bit running late, so the discussion time is limited. But for all the unanswered question can be done by email after this webinar. We probably go with the first one, developing PK models to inform dosing traditionally required several consecutive blood samples to be taken from each patient in order to describe the plasma drug concentration versus time curve. That's not always possible, especially for young and sick patients. Do you have any advice?
So I'm guessing the question refers to pediatric drug developments, right? So normally nowadays in drug developments, you always start off developing a drug using adult populations, right? So once you finish adults, then you would eventually move on to pediatrics patients. So normally you would collect a lot of samples. So you have dense PK samples in adults. So then you will have a population PK model built. Then eventually you move on to adolescents, and then you move on adolescent subpopulations. Then you move on to pediatrics, so people of a younger age. So by doing so, essentially you are leveraging information that you collect in adults. So what we normally do is we have adult models, then we have adolescent, and we have the adult models. Then we use those adult models to design your studies for your pediatric studies. Then that you can select the, how many samples, sampling point samples you need, as well as the sampling time, as well as the sample size. And from there, you would then start your pediatric drug development. So firstly, usually start out with adolescents. So you adult, and then you combine, you would merge your adolescent data with your adult models, and you would update your models, and then to predict the next cohort. And then if the model described it well, then you would, you would then continue to use that model, or you would redevelop or refine the models. If the prediction is completely off, then what you do is you refine your models, just like the examples I shared early on. Then this is essentially an iterative stage. So by doing so, you do not need to collect crazy amount of samples in pediatric, because there's a restricted volumes that you could collect from a very, very young children's. But if you follow the approach that I just mentioned, essentially, you do not need to collect a huge amount of samples per subject in the pediatrics. And we will have a talk from a well expert, in pediatrics. She will be giving us a talk on, pharmacometrics in special populations, and pediatrics will be one of the topics. And she will be giving you examples of how pharmacometrics can be used to optimize clinical trials for pediatric clinical trials.
And from Stephen, in the triple combo example, can the characteristics of patients likely to respond be determined so that only they receive the treatment, and can patients who won't respond be identified and assigned to an altered treatment?
So in a triple combination example, can the characteristic of patients likely to respond to be determined so that they only receive the treatment, and can be patient who? So the examples I show you today are essentially translational models. So we developed the PK/PD model in test tubes, and then we link those to human PK profiles, and to optimize the therapy, or to select the first in human dose. So if you have, let's say if you are in a phase 2 or phase 3 clinical studies, right, and you have collected the data, obviously in clinical trials, you won't be able to collect the bacterial count across times. So normally you have some sort of an efficacy endpoint. So you will be either response to the therapy or not response to the therapy. And in that particular case, you could still do pharmacometric models, but not, to describe those relationships. And you could potentially still do covariate modeling to see what are the likely factors in addition to concentrations in causing those patients to respond or not respond to your antibiotic therapy. So I guess the question is, yes, you can do that, but not with the models that I showed today, but there are other models that we could use, techniques that we could use to do this.
Thanks. So in the interest of the time, we probably just go the following 2 questions, and then for the all the unanswered question, we can go offline. The next one is the triple therapy. Do you actually conduct RCT in 10,000 critical ill patients?
No. So what we actually did is virtual clinical trial simulation. So we remember in the population PK models, we characterize the dose and concentrations relationship. So we know when you give a particular dose, that will give you a certain concentration time profile, right? But if you change the dose, the concentration time profile will different, right? But if you give the same dose to me or Jason, the exact same dose, our PK profile will be different, right? So exactly what the population PK modeling does, it captured the variability among different individuals. So we know that shape is normal distributions. We know the spread of the distributions of clearance, for example. Let's say there's a 20% variations for clearance for polymyxin B. That's not true. That's just an example. And therefore, we can sample from that distributions. So we can, and then we create a virtual populations. So then we essentially, what we do for the triple combination therapy is we did a virtual clinical trial simulations, try different dosing regimens, and we find, okay, this particular dose is most effective against this particular strain, but not very effective against another strain. So then we can use that dose to move on to RCT experiments, move on to clinical trials. So rather than doing dose finding in humans, we have already selected a few optimal, few best doses in our models, and then we can maximize the success rate of our clinical trials. So essentially, you're not doing guesswork. So a lot of time when you start off the clinical trials, you are guessing or you're not too sure. There's some sort of a guessing exercise going on because you don't know what dose you should select. But with model exercise, you have a scientific rationale or a scientifically designed starting dose for you to do in your clinical trials. That would then, of course, increase the success rate, decrease the cost, and decrease the failure rate. I hope that answered your question.
Yeah, thanks. And the last one, it shouldn't be a question. Like Leo actually interested in antimicrobial PKPD modeling and following a master of. Yeah, do you have any internship opportunity?
Yeah, so Leo, our group at the the Malaya Translationals and Clinical Pharmacometry groups do have an internship positions available, and we do have weekly workshop as well on pharmacometrics. So if you're interested, please just you should have my LinkedIn as well. So please just send me a message, and I'm happy to talk to you, and we're happy to include you in our group as well. Thank you.
Thanks, Dr. Yu-Wei. I think in terms of the time, we probably finished here. And if anyone has unanswered question and for the question, feel free to drop email or follow our social media to reach out. Thank you.